Good afternoon, everyone. Welcome to the CET NEET coaching. So today we are starting our regular classes, and this is the biology class, and the chapter is molecular basis of inheritance. So now we'll be discussing 30 questions and then the answers of it. And apart from that, in the question paper set, I have given 15 extra questions for you to practice. So these questions we have done in the trial class, but we'll go through it quickly once again. So the first question is, a series of experiments A series of experiments were conducted by Frederick Griffith in 1928 on transforming principle with which microorganism? So you just have to look at the key terms here. That is, you have to look which is the scientist. It is Frederick Griffith, and on which did he do? This was the transforming principle experiment. So transforming principle experiment was done by Frederick Griffith. In which organism? It was done in Streptococcus pneumoniae. Pneumonia is the disease that is infecting the lungs, and this is the organism which infects the lungs and causes pneumonia. And this experiment we had discussed earlier. So this is the answer for this first question, Streptococcus pneumoniae. It was not in Escherichia coli. It was not in Bacillus thuringiensis. It was not in Salmonella typhimurium. So the correct answer is Streptococcus pneumoniae. So this was the experiment. He injected the Streptococcus pneumoniae bacteria into rats, and the rats got pneumonia and it died. So which was the organism? It was Streptococcus pneumoniae. This experiment we had discussed earlier. So now we'll go on quickly to the next experiment. Next question. So the first answer is option A, Streptococcus pneumonia. Okay. The next question, in lac-operon concept of gene expression, allolactose act as. So lac-operon concept again we had learned, but we'll just quickly go through it. This is the lac-operon concept. Here you can see, if the lactose is present in the media for the bacteria to grow, then the bacteria will, if lactose and glucose is present in the media, the bacteria likes and bacteria loves glucose. It will eat on glucose. It will feed on glucose. But if glucose is absent in the media, then the bacteria will start surviving by eating lactose. But if it has to eat lactose, it has to produce some enzymes which will break down lactose. So here we have two cases. Okay, if lactose is present in the media, if lactose is absent in the media. In the first case, if lactose is absent in the media, I'm sorry, one second. Yeah. If lactose is absent in the media, if there is no lactose, so the bacteria need not produce any enzymes to break down lactose. Okay, so there is a repressor produced by the DNA, and this repressor will block the production of enzymes for breakdown of lactose. But if lactose is present in the media and the bacteria wants to utilize this lactose, then somebody will block this repressor. Somebody will block this repressor from going here, and that is called the inducer. And here the inducer is the lactose molecule itself. The lactose molecule will block the repressor from blocking the DNA to produce the enzymes required for the breakdown of lactose. So there is lactose in the media, and the lactose has to be broken down with the help of some enzymes. So some of the lactose will act as an inducer. It will block the repressor. So the answer for this question is, who acts as in the lac-operon, who is acting, what is the action of allolactose? Allolactose is the inducer. When it is acting as an inducer, the lactose molecule is also called allolactose. If lactose is acting as the inducer, it is called allolactose. Okay. So now we go on to the next question. The number of codons effective in coding 20 amino acids. We know that we have for protein coding enzymes, or for protein coding, we have 20 amino acids. But we also have some codons in the mRNA which will code for the amino acid. What are codons? These are 3-letter codes which will code for one amino acids. So here we have a chart. Here 3-letter codon codes for different amino acids. This chart is there in your textbook, so you must have seen this. So now how many codons are actually useful for making amino acids? This chart has 64 codons, but out of this, 3 are not used for making amino acids. 3 are called stop codons. 3 are called stop codons. You can see here 1, 2, 3. So 65 minus 3. So you have only 61 codons are useful for producing amino acids. Okay. So number of codons effective. How many codons are effective in coding amino acids? It is just 61. So next question. Which aspects form the basis of DNA fingerprinting? So what is DNA fingerprinting? Every person has a fingerprint. In the same way, every individual fingerprint, but there is a chance you can, do some plastic surgery and change it. Some criminals do that, or sometimes there's a very rare chance that one person's same fingerprint will be there for another person. But inside your DNA also there is a print which you cannot change. That is called DNA fingerprint. So my fingerprint will be different from your fingerprint. So you can use the fingerprint to see who with this. Suppose you collect from a crime scene. If you collect some blood sample, or if you collect some hairs or something like that, if you want, you can check whose is that hair or whose is that particular body fluid. You can check that because that will have the print inside the cells. So this is called DNA fingerprinting. So what is the basis of DNA fingerprinting? So if you look here, this is the DNA from one individual B, and this is the DNA from another individual A. So if you break inside the cell, inside the nucleus, the DNA is there. If you fragment it, and then if you run it through a gel, then you will get like these bands. So this is the band for one individual. This is the band for another individual. Now you have these two bands. Now there is another band which you want to confirm whose is it. Suppose this is a criminal's band. So if you want to check, this is suppose for some we can say individual A, this is individual B. So you look which is matching. So when you look here, you can see clearly. You can see clearly this band is matching with this one. So actually this criminal is this person. So like that you can match. So what is actually the DNA is being broken down into pieces and run through a gel. So what is the basis of this DNA fingerprinting? And the answer is satellite DNA. So the colorful bands that you see here, see, these colorful bands that you see here, that is called satellite DNA, or it is also called VNTR, or it is also called satellite DNA. It is a repeated sequence of bases, nitrogen bases. It keeps on repeating, and the number of that repeating unit will be different for different individuals. So that you are comparing between one crime scene or the suspect or whoever you want to check. Sometimes you do this for paternity checking. You check the DNA of the child and check the DNA of the two fathers. So with with which father the child's DNA is matching, so this child will be their kid. So paternity tests or for criminal crime scenes, you can use DNA fingerprinting. So there is a repeated sequence of nitrogen bases that is repeating. It is called VNTR. So that is the basis of DNA fingerprinting. It is the satellite DNA, VNTR, showing high degree of repetition. It keeps on repeating in DNA segment. This is the basis of DNA fingerprinting. This is wrong. Amount of DNA in the sample. Amount of DNA is not the basis of DNA fingerprinting. Ratio of purine and pyrimidine, that is not the basis. Okay, the sequence present in the ridge and groove, that is not the basis. Correct answer is option D. Now the next question, what is the anticodon found on the tRNA for tryptophan amino acid? Okay, so you have to find out what is the anticodon. Again, you come back to the same chart. You know your chart. Okay, so in the chart, UGG shows that UGG codes for tryptophan. You can look into your textbook. In that 64 amino acid codon chart, you have UGG codes for amino acid, tryptophan. But the question here, what is the anticodon? They are not asking you the codon, they are asking you the anticodon. So this is the chart, and if you look here, you can see UGG coding for tryptophan. See, UGG codes for tryptophan. Okay, tryptophan amino acid is coded only by UGG. But the question is, what is the anticodon? So what you have to do, you look here. You know that mRNA, this is the mRNA. mRNA is first going inside the ribosome. This is the ribosome. And then you can see tRNA is coming with amino acids. Different tRNA is coming with amino acids, and the tRNA will read the codon here, and then the matching tRNA will bind here. So this codon will bind with the complementary tRNA, and that amino acid will be put here. So the amino acids will join to form a big chain, and that will become the protein. So first what happens, the mRNA will go inside the ribosomes, and the tRNA will come with the amino acid, and it will check on the mRNA what is the code written. Okay. So what is the code written here? That will be read by the tRNA, and the tRNA has an anticodon. And the mRNA has a codon. So codon and anticodon will be complementary base pairing with each other. So the codon here is UGG. If the codon on the mRNA is UGG, then the tRNA will have anticodon. What will be complementary to UGG? U complements with A, G complement with C, G complement with C. So this is the tryptophan amino acid. So what is the anticodon? The answer is ACC. Okay, so the answer is ACC. What is the anticodon? Anticodon for tryptophan amino acid is ACC. So first you should know what is the codon for tryptophan, and you should try to remember. You cannot remember all the 61 codons, but try to remember for methionine and tryptophan, because they have just one codon each. Okay, methionine it is AUG, and for tryptophan it is UGG. Try to remember these two things. This is for methionine, and this is for tryptophan. These two at least you should remember. Okay, so now we are going to the next question. DNA replicates semi-conservatively was first shown in which organism? So you know that DNA replicates in a semi-conservation fashion. So if this is the parental DNA, when you open this, then the new daughter DNA will be synthesized. Okay. The new daughter DNA will be synthesized here. And now when you give this to the two daughter, one daughter will have one parental DNA and one daughter DNA. The other cell will also have one parental DNA, one daughter DNA. So you are only half of the parental DNA is conserved. That is why it is called semi-conservation model. This was first proved in which organism? It was proved E. coli. So the answer is E. coli. This is the semi-conservatory model. This is the semi-conservatory model. This is not the model that we follow. Only this is the model that we follow, semi-conservatory model. Okay, in which the parental strand, only half of it is retained in the daughter strands. So this was proved in E. coli. In which experiment? I told you the blender experiment. This is the blender experiment in which this was done by which scientist? Hershey and Chase. So you should know the name of the scientist as well as the experiment. And it was done in which microorganism? It was done in E. coli. So the answer is E. coli. After that, later on, it was also done in plants. But now first, the question is, it was first shown in which organism? First, it was shown in E. coli. Okay, so you should read the question carefully. So can we go to the next question? Polymerization of DNA nucleotides during the synthesis of lagging strand occurs in which direction? Okay, so DNA will always be synthesized, DNA or RNA, everything will always be synthesized from 5' to 3' direction only. So this is the correct answer. Only in this direction. DNA cannot be synthesized in the opposite direction. So this is to confuse you, that is wrong. Okay. So this is wrong, and any direction that is wrong. Promoter to terminator direction, that is not specific. Okay, this is the correct answer, 5' to 3' direction. See, if you see here, always from 5' to 3', 5' to 3'. Okay, it is always in that direction. This is the 5' and new nucleotides will join here. So if this is the starting of the 5', the chain will grow in this direction. Chain will grow in this direction. That is why it is called 5' to 3' direction. So that is the correct answer, 5' to 3' direction. That is the odd sequence in which the nucleotides are added. Okay, now the next question is the length of DNA helix in a typical nucleosome. So if you take a nucleosome, I told you last time, if you look into the DNA, it is if you look into the cell inside the nucleus, DNA is found in the shape of a chromosome. Okay, but actually the chromosome, if you unwind it, you can see the DNA. The DNA is actually packed around some small beads, like like a necklace, so many beads in a necklace. In the same way, the DNA is packed around small beads, and these beads are called nucleosomes. Okay, they are made up of a protein called histone. These are the small beads around which the DNA is wound. Okay, and that beads are made up of a protein, special protein. That protein's name is histone. Okay, and then when it is wrapped around this whole structure, it is called nucleosome. Okay, so this is how the DNA is packed. So the DNA is packed around the nucleosome, and the nucleosomes are packed to form the chromosome. Okay, so the length of the DNA around a typical nucleosome. If you have one nucleosome, how much of DNA is around that? That is about 200 base pairs. So the answer is 200 base pair. Base pair, why are we not saying centimeter, meter? Because the length of the DNA around a small nucleosome, that will be very small, right? So you cannot measure it in centimeters or meters. So you will say how many bases are there, how many nitrogen bases are there, and nitrogen bases occur in pairs. So you say 200 base pairs. Okay. So the next question. Which of the following type of RNA carries amino acids towards the ribosome during translation? So we have discussed about protein synthesis. In protein synthesis, there are two steps, transcription and translation. So you have transcription, which is happening inside the nucleus, in which the DNA will give you a small copy of mRNA. DNA will give you a small copy of mRNA. Then this mRNA will come out of the nucleus. This is happening inside the nucleus, where mRNA will come out, and then ribosomes will sit on it, and then ribosomes will make the proteins. This is called translation. Okay. Now, who is bringing amino acids to the ribosomes? So, tRNA carry the amino acids. So, which of the following type of RNA carry the amino acid? Who is carrying the amino acid? Who is carrying the amino acids? Who is carrying the amino acids? The answer is tRNA. tRNA is carrying the amino acids to the ribosome. This is the ribosome. This is the mRNA. So proteins are synthesized here. So tRNA is carrying the ribosomes, amino acids. Okay. So now we'll go on to the next question. In eukaryotes, the entire base sequence of the gene does not appear in the mature RNA. So you know that, yeah. So if you look into a eukaryotic cell, you have the whole DNA. Now you need only a particular small part of one gene if you want to copy into make a protein. First, from that gene, you have to make an mRNA. If this is the if this is the gene, so if this is the gene of your interest, you copy this to make an mRNA. This is the mRNA. But this mRNA will also have some unwanted parts. That is called intron. If this is the mRNA, you have some unwanted parts called introns that should be removed. You don't want, only want these two. So this is removed by some proteins. Always whoever is doing job inside the cell, they are proteins. Proteins are the molecule which do some work inside the cell. So who will remove the intron? Another protein. Okay. So these green proteins, I'm not telling the name, they come here and they remove the yellow part. You don't need the introns. Okay. If you want to know the name, these are SNRPs. SNRPs will come and remove the introns. And then only what is required, they will be joined together. This is the processed final mRNA. Okay, so this mRNA is not what you require. You require only this. When you copy the gene, when you copy information into mRNA, so many unwanted things also come. So that has to be removed. This is called splicing. This is called splicing. Okay, this is called splicing. The protein is sometimes referred to as spliceosome, and then you are removing the intron. Some of the terms you should know. So what are you removing? You are removing the intron. What do you need? You need only exons. There is an exon here, there is an exon here. You need only the exons. So intron you don't need. Intron you are removing. And the removing proteins are called spliceosomes or SNRPs. Okay. So in eukaryotes, in prokaryotes, this doesn't take place. This takes place only in eukaryotes. In eukaryotes, the entire sequence of gene is not seen in the mature RNA. Mature RNA is the final product. In the final RNA, you cannot see all the sequence because some of the sequence are removed. Some of the sequence are removed. Why? Introns are removed during processing. When you process, see, when you have some, you just compare it with noodles. When you have wheat or something in your field, that is the raw food. But now you process it, you remove so many things, process it and break it down, and then finally this becomes noodles. So there is a processing happening. In the same way, when you look at noodles, you will not see the wheat grain. Okay. In the same way, when you initially you have some RNA, but when you do the processing, you remove all unwanted stuff. Introns are removed by processing, and then you get the final mature RNA. This is called mature RNA. Then you have removed the introns. Now let's go to the next question. Suppose DNA samples are collected for DNA fingerprinting analysis, which are less in quantity. Suppose you go to a crime scene and then you are trying to find out something so that you can check who did the crime. Okay, but when you look around, you are not getting anything. So finally you got only one or two hair follicles, or some body fluid you got, some saliva or something you got. If it is very little, then you will get only very little DNA. Then you cannot analyze. It is very difficult to analyze who is the criminal. So you have to increase the amount of DNA. For that, what machine, what technique you use? The answer is PCR. What is the machine or the technique? PCR. What is the full form? Polymerase chain reaction. Polymerase chain reaction. Polymerase chain reaction. Okay, so that is the technique that you use to increase the amount of DNA. You will not use all these things. Chromatography, no. Chromatography is a technique used to separate the components present in a mixture. So it is not chromatography. Electrophoresis, you apply electric current to separate the proteins or separate the fragments of nucleic acid. So that is not. DNA probing is a technique used to find out your special sequence of interest. So the correct answer is polymerase chain reaction, PCR. Okay, so this is the reaction. Here, if you have just one piece of DNA, if you put it into that machine, you will get millions of copies of that DNA. After some time, you will get millions of copies, just like a Xerox machine. You will get so many copies. So this is called PCR, polymerase chain reaction. So let's go on to the next question. When Escherichia coli cells are cultured in a medium where lactose is absent. So E. coli is also called Escherichia coli. These cells are grown in a medium. You're growing it in a plate or you're growing it in a flask. And there you have, there is no lactose. Okay, there is no lactose. But the cells are growing. Maybe there is some glucose. Okay, then the I gene of lac operon continues to produce repressor mRNA because. So what is this? In the lac operon, yeah, when Escherichia coli is grown, even though lactose is not there, the gene responsible for producing the enzymes to break down lactose, that gene is producing repressor. So why is repressor still produced? Repressor is always produced because if there is no need of producing the lactose breaking down enzymes, but still you have to produce, the cell will produce repressor. If something is always produced, it is called constitutive gene. Okay, if the gene of lac operon continues to produce, it is always produced. If something is always produced, then it is called constitutive gene, because this repressor will go and block the gene from producing enzymes. We have discussed lac operon before. So now we'll go to the next question. For the given sequence of DNA, identify the complementary sequence of bases. Now you have to make the complementary sequence of this. This is a very easy question, right? On its mRNA from the options given below. So look here. If this is the sequence, how to write the complementary sequence? If it is A, then you have to write U. If it is T, you have to write A. If it is G, G is complementary with what? C. Okay, C is you should just remember this rule. A will always complement with T and T will complement with A. G will complement with C and C will complement with G. So if you know this much, you can write that. If it is A there, you write T here. If it is G there, write C here. Okay. So if it is A, you write U. If it is T, you write A. Why am I not writing T? Because you have to write for RNA. If RNA, there is no T. So you have to remove T and write U. Okay. So if it is A, you write U. If it is T, you write A. If it is G, you write C. If it is C, you write G. If it is again A, you write A. So no need to write the whole thing. Just now look, UACGA. UACG. Okay. And one thing you should remember, there are two things which is showing the same. But now you should check for this side. If it is 3 prime here, then in the answer you will be having 5 prime. Because the 2 nucleic acid is always put in anti-parallel direction. Understood? So this is how you find the answer. Now let's go to the next question. In prokaryotes, transcription of DNA is initiated with the help of which protein? So in prokaryotes, there is a special protein that helps in transcription. You know, transcription means making RNA. So RNA is made with which protein? RNA polymerase. But in prokaryotes, RNA polymerase is associated with a small unit that is called sigma factor. In prokaryotes, RNA polymerase is associated with a small important factor called sigma factor. So sigma factor, this is only for prokaryotes, means microbes. Okay. In prokaryotes, in eukaryotes, you don't have sigma factor. Sigma factor or sigma subunit is very important. Okay. So in prokaryotes, transcription you need with the help of which one? So you need sigma factor. Initiating sigma factor is helping to start the protein synthesis, start the mRNA synthesis. That is why if it is initiation, you should write sigma factor. And if it is termination, if you want to stop the synthesis, what is the factor that is helping in prokaryotes? It is Rho factor. This is for stopping, for termination. Stopping is also called termination. Okay. For stopping, you need Rho factor. For starting, you need sigma factor. Okay. And then elongation factors, it will help to elongate. And termination factor, you need not say, it is the same as Rho factor. Okay. Yeah. Now we are going to the next question. According to Human Genome Project, okay, total number of genes in human genome is 30,000. So in the total human gene, you have around 30,000 genes. But they are asking you the genes present on Y chromosome. The question is only here. The question is only this much. The number of genes present in Y chromosome. How many genes are present in Y chromosome? So if you look here, this is the karyotype. This shows the total number of chromosome inside a human cell. Inside human cell, you have 46 chromosomes. And actually, there are 23 types. And first, each one is in pair. So you have 23 pairs. Okay. So this is the first pair. This is the second pair. In the human cell, you have DNA. The DNA is actually in the form of chromosomes. And you have 23 pairs. You have 23 pairs. Okay. This is the first pair, second pair, third pair, fourth pair, fifth pair, sixth pair, seventh pair. All are in different sizes. This is a special pair. This is called the sex chromosomes. Okay. This has X and Y. And you can see Y chromosome is the smallest chromosome. So it will have the least number of genes. If you look here, see, this is the number of genes present in Y chromosome. Y chromosome has very little amount of genes because its size is very small. So it is 231. Look here, Y chromosome is the smallest chromosome. It has only 231 genes. Okay. And chromosome 1 is the biggest chromosome, so it will have the maximum number of genes. Look here, chromosome 1 is the biggest number of, biggest size, largest chromosome. So it will have maximum number of genes. Now coming to the next question. Again, in a crime investigation scene, the investigating officer collects different samples from the crime spot for DNA fingerprinting. Which of the following sample is not helpful? Which is not helpful? So from the scene, he is collecting different samples. He is collecting some skin shreds. Skin shreds means, what is skin shred? That is the skin. Every day from your skin, lot of cells fall down. When you clean the floor, you get a lot of dust. All these are, most of it, 60% is your skin cells. So there will be skin cells, there will be erythrocytes, blood cells. So he collected skin cells. Okay, skin cells. Erythrocytes means blood cells. Blood cells, he collected semen. Semen means sperm cells. Then he collected some hair follicles, some hair. So which is not useful? This is useful because hair, hair follicles means it will have some cell. Follicle means the root of the hair. So it will have cells and nucleus and DNA. And sperms will have nucleus and DNA. Okay, skin will have cell, nucleus and DNA, but blood sample is not useful. Erythrocyte inside the blood is not useful. Inside the blood, there are different types of cells, but erythrocyte means RBC. Erythrocyte means RBC. Red blood cell. Red blood cells have, what is the function of red blood cell? It will carry oxygen. It has hemoglobin. It will carry oxygen. It has hemoglobin. So it is not having nucleus. The mature RBC is not having nucleus. So there is no DNA. Nucleus is not there. There is no DNA. So you will, if there is no DNA, you cannot use it for DNA fingerprinting. So the correct answer, which sample is not used? You cannot use RBC because RBC, mature form of RBC, does not have any nucleus or DNA. Now the next question is, a mature mRNA, mature means the final mRNA after processing, consists of 900 bases. Okay, there is no stop without any stop codon in between. Calculate the number of amino acids coded by the mRNA during translation. So suppose you have, so one mRNA has 900 bases. So after every three bases will code for one amino acid. So if you have 900 bases, how many amino acids will be there? That is the question. So 900, sorry. Yeah. So you have 900 bases. Every three bases will code for one amino acid. So you have again the next three will code for the second amino acid. So 900 divided by 3. That is how much? 300 amino acids. So the answer is 300 amino acids because every 3 amino acids will code for, every 3 bases will code for one amino acid. So that was simple max. Now let's go on to the next question. This is from that codon chart. The codons on mRNA are so and so. If this is the codon, which will be the amino acids? So CAU, you should remember from the chart. You can look into your chart now, but you should remember later on. CAU codes for which amino acid? CCU codes for which amino acid? AAA codes for which amino acid? CUG codes for which amino acid? So you should remember that. CAU, see, CAU codes for leucine. So what, if you at least remember one thing, you can check in this. CAU, it codes for leucine. But there are two leucines here. Okay, next is CCU. What is CCU? CCU codes for proline. CCU codes for proline. Okay, then you have AAA. AAA codes for which amino acid? AAA codes for lysine. So this is the first answer is the correct one. Okay, AAA codes for lysine. CUG. Again CUG. CUG. CUG codes for leucine. So you should remember this chart. The whole chart is difficult to remember. At least remember these things. So at least these many amino acids you should try to remember so that you can correlate. If they give you the codon, you can say which is the amino acid. So the correct answer is option A. Now we go to the next question. Again, they have given you the amino acids and they are asking you to find the codon. But this is a bit more, you can see methionine, it is always AUG. So you cannot use that. If you know methionine, you cannot find the answer because AUG is present in all the options. Then you have to look for leucine. So as we have seen here, leucine is coded by many amino acids. So if you can remember that, then only you will be able to answer this question. So if you look here, the correct answer is DAUG, CUA, GUG, CGU, and GCC. That's the last option here, option D. Now we'll go to the next question. Sickle cell anemia is caused. This is a very important question. It frequently comes in the exam. This is a disease called sickle cell anemia in which the blood cells or the erythrocytes, the RBCs inside your blood will form a sickle shape. It will be sickle shaped. It will have the shape of half moon or sickle shape. Okay, so that is why it is called sickle cell anemia. And these RBCs cannot carry oxygen that much, so the people will have difficulty to breathe and they will be suffering from anemia or hemoglobin deficiency. So in sickle cell anemia, what is the change that is happening in the DNA? If you look carefully, this is a normal person. For the normal person, the DNA codes for, this is the sequence of the gene. Okay, and this is copied to form the mRNA, and the mRNA will be copied to form the amino acid, and other amino acids are also here, and together it will form the protein. But what happens in some people, there is some genetic mutation. What is mutation? There will be some change in the sequence. So instead of A, here you have, instead of A, you have T here. So T will base, means A will pair with T, but here you have T pairing with A. Because of this mutation, instead of amino acid GLU, you are getting VAL. Instead of glutamic acid, you are getting valine. So that is why the hemoglobin will be deformed and the RBC will be having a sickle shape. Sickle shape means like a C. It is not properly round. The RBC will be semi-round in a sickle shape. Okay. So what is the change that is happening here? From CTC, it is becoming CAC. Or you can say GAG is becoming GTG. Whichever you find, you can write the answer. So here, mutated G is CTC is becoming CAC. CTC is becoming CAC. So that is the answer. So this you should remember always in sickle cell anemia, CTC is becoming CAC. Okay, next question. In the given transcription unit, identify the regions 1 and 2. This is region 1 and this is region 2. This is region 1, this is region 2. So you should say which is which. Now, if you look at this picture, it is difficult to understand, but they have written here template strand. So the template strand, I told you always the synthesis happens in 5' to 3' direction. So if suppose the synthesis has to happen in 5' to 3' direction, it will not happen here. Because if you write here 5, this is again 5. That is not the correct way. You should write always in the anti-parallel fashion. So what should you write? This is 5' to 3' synthesis will happen from here. From here, this is 5' and it will start synthesizing in this direction. Okay, so if synthesis is starting here, this is the promoter. Promoter is always in where? Promoter is seen in the starting and terminator is seen in the ending. Okay, so with respect to template strand, when the synthesis is starting like this, the starting point is called promoter and the ending point is called terminator. Okay, so if this black lines are the DNA and this red line which I drew is the mRNA which is formed. This is transcription happening here. Transcription is copying the mRNA from the DNA. The black lines are the DNA and the mRNA is being copied from the DNA and it is synthesized in 5' to 3' direction. So it is starting from here because 3' is here, so 5' should be here. Then only it will become anti-parallel. So it is starting in this direction. So this is the second thing is promoter and the first thing that they mentioned is terminator. Okay, so the answer is, if they ask, first one is terminator, second one is promoter. So first one is terminator, second one is promoter. According to the question, first one is terminator, second one is promoter. But if you look into the DNA, the synthesizing starting area is called the promoter. So what we have written is correct. So this is the promoter and this is the terminator. So they asked, first you tell this, then you tell this. So your answer is terminator and promoter. Now, which of the following sequences of mRNA are required for translation process which are not translated? So if you look at the typical mRNA, one mRNA has many parts. It has a 5' cap. mRNA has a 5' cap. In the 5' end, it has a cap. Then in the 3' end, it has a tail. It's called tail. Then this is the important part of mRNA. This is coding for proteins. These has the codons. Codons, which will give protein. Okay. Then here you have UTR. This is called 5' UTR. UTR present in the 5' area. Again, here also you have UTR. UTR present in the 3' area. What is UTR? It is untranslated region. What is UTR? Untranslated region. Means they are not translated. In the second step, protein synthesis has two steps. First is transcription, second is translation. In the second process, translation process, this is not useful. That is why it is called the untranslated region. Okay, so what is not useful during translation? Which of the following sequences are required for but they are not translated? Which are not translated? That is UTR. So if you look at some keywords, you will be able to answer. In the mRNA, sequences of mRNA, sequences of RNA which are not translated, they are UTR. Okay. And the next question, DNA present inside the nucleus was named. Who found? It was Friedrich Mister. So this scientist, Friedrich Mister, he found out that inside the nucleus something is there and he actually named it. It was found by somebody else, but he gave this name, nuclein, nucleus, and nuclein was given by the name was given by Friedrich Mister. So it was Friedrich Mister who gave the name nuclein. And Watson and Crick, you know, these are the scientists who gave who proposed the double helical model of DNA. Okay, and Rosalind Franklin, she took the photograph of the DNA, X-ray photograph of the DNA for the first time. Okay, and he also worked with them for producing the double helical model of DNA. So the answer is Friedrich Wischer. Now next question, when does lac operon in E. coli become switched on? Lac operon is coming again and again, it's an important thing. So when is it becoming switched on? Switch on means it is producing the enzymes required for the breakdown of lactose sugar. Lac operon is a gene, or it's a group of genes which will produce enzymes which will help in breakdown of lactose. If you are eating something, it should be broken down to give you energy. So the microbes, if they want to eat lactose, they should have some enzymes to break down lactose and take energy. So if the gene is producing the enzyme, it is called switch on. Okay, if the gene is not producing the enzyme, it is called switch off. Okay, and this repressor molecule is always produced. That is why it is called the constitutively produced. In another question you saw, constitutively. Constitutively means it is always produced. Okay, so repressor is somebody, it's like a gate. It will block the synthesis of enzymes if it is not required. But if you want to switch it on, then this security repressor is like a security guard. It should be blocked. That will be blocked by inducer. Inducer is nothing but the lactose molecule itself, and that time you call this inducer allolactose. Okay, you call this allolactose. Now when question here is, when is it switched on? When is the lactose operon switched on? When lactose is present in the medium, then lactose operon should be switched on. Okay, when it is present in the medium, it will act like an inducer and it will bind to the repressor. Repressor is like a security guard. It will go and block the synthesis of enzymes if it is not required. So what will the lactose molecule do? Lactose molecule bind to the repressor and tell him, please don't stop the synthesis. We have to make the enzyme. So when lactose is doing that, lactose is called allolactose or inducer. So this is the correct option. Okay. So when does lac operon become switched on? When lactose is present in the medium and the lactose will go and bind to the repressor and tell him, please don't block the synthesis. Okay. Now the next question, in which of the following steps in DNA fingerprinting technique are labeled VNTR probes used? So when do you use probes in DNA fingerprinting? So you should know the steps. Then only you can tell when you use the probes. So let's see the steps in DNA fingerprinting. So this we have talked about. So suppose you have some saliva or some sample from the crime scene. So what you have to do is you take the DNA and you break it down into pieces. Then you run it through a gel. You are running it through a gel to separate the pieces. Okay. So now you separate the pieces and then you will put some probes. Probes are small molecules which will help to identify your gene, whatever you are looking for. It's like a torch. You can see what you want to find out. So when do you use probes? You use it in the last step. So here you are using probes to identify. Then you see what you want to see. It's like a torch to identify what you want to see. So during hybridization, during the last step, so these probes are having small light. So they will go and bind to the DNA, pieces of DNA of your interest. What are the pieces of DNA of your interest? These are the pieces of DNA of your interest. If you want to see these DNA, actually DNA is not colored. You cannot see this red, green, blue color in the DNA. You need some, it will be all white. So if you want to see that, you need some torchlight. So this torchlight is, you have some molecules with a small color emitting chemicals. So these are called probes. So this should go and bind to your DNA. It should go and bind to your DNA. Then only your DNA will be clear. So then only your DNA will be visible like this. Okay. So this binding is called hybridization. This binding is called hybridization. So these torchlight things should go and bind to the DNA. That is called hybridization. Then you can see it very clearly. The bands. Okay. So when is this probes used? Probes are the torchlight like things. When is it used? During hybridization. Now you have to know the number of nucleotides, number of base pair present in some, at least in few organisms mentioned in your textbook. Okay. So what is the correct answer? So how many nucleotides are present in E. coli? How many are present in humans? How many are present in Drosophila? You should know. Match the number of nucleotides of the genome of column 1 with organism in column 2. So you can see here, this is column 1 and this is column 2. Here you have the number of nucleotides. Here you have the organism. So this is the smallest number of nucleotides. This goes to bacteriophage lambda. Okay, bacteriophage lambda. Bacteriophage is a small virus which eats bacteria. Next is this. This is also another bacteriophage that is called 5x174 bacteriophage. That has 5,000, about 5,000 nucleotides. Now, when you go to viruses are very small, so they have 5,000 or 4,000, 48,000, like that nucleotides. Okay. Now you have this big amount of DNA. That will be obviously for human beings, right? 4.6 into 10. This is highest is 10. Look at the power to it which it is raised. 10 raised to 9. 10 raised to 9 is the highest. So this is for human beings. And this is for E. coli. 3P. This is for E. coli. Okay. So this is the smallest, right? 4,386 is the smallest. This is for this. This is for this. This is for human beings. And this is for E. coli. So the correct option is what? Correct option is C. 1S, 2T, 3P, 4Q. So the correct option is this one. Okay. Now the next question. What is the average length of hnRNA in humans? hnRNA is the unprocessed RNA. Before, as soon as the RNA is produced after transcription, it is not processed yet. It is not having a cap. It is not having a tail. It is not having UTR. I mean, it is not having other introns are not removed. So it is having a lot of unwanted junk material. So it is a very long RNA. So the length will be really very big. So what is the average length of? It is 3,000 bases. But then when you remove, spliceosome comes and it will remove the unwanted introns. It will give it a cap. It will give it a tail. Then the length will reduce. Okay. Yeah. Now the next question. If E. coli is allowed to grow for 40 minutes in a medium containing nitrogen 15. Nitrogen 15 is heavy nitrogen. Nitrogen 15 is a heavy atom, heavy nitrogen. Then the number of DNA containing nitrogen 14. This is a very simple and silly question, because the E. coli is grown in nitrogen 15 medium, but they are asking you how the amount of DNA containing nitrogen 14. So there won't be nitrogen 14 in that, because you have grown 40 minutes in N15. So the answer would be 0. Okay. Yeah. The next question is, gel electrophoresis is used for what? So this is the second last question. Gel electrophoresis is a technique. It is used to separate the DNA molecules. So what is the correct answer? Is it used for cutting the DNA fragment? No, it is not for use for cutting. Is it used for constructing recombinant DNA? Are you recombining DNA of different organisms? No. Then is it used for isolating DNA molecules? No. The correct answer is it is used for separation of DNA fragments according to their size. Okay, so this is the setup. This is a gel electrophoresis machine. So in this tray you have a gel. Now when you give the DNA, if you put DNA here, the DNA, if you put DNA is having a lot of, if you cut the DNA initially and put it into the gel, then different sizes will come out like different bands. This is called gel electrophoresis. So what is it used for? Separating the DNA according to different sizes. Okay, so the correct answer is separation of DNA fragments according to their size. This is the last question. The given diagram represents. So this is a simple diagram from your textbook. You have to identify the diagram. What is this? Is it a mesosome? Is it a chromosome? Is it a ribosome? It is a nucleosome. What is the correct answer? I'm sure you all know it. It is a nucleosome, because here it is written histone DNA. So histone proteins are present in nucleosome. Ribosome, what is ribosome? Ribosome is the protein producing factory. Ribosomes, through which the mRNA goes and proteins are formed. So it is not ribosome. Chromosome, you know, chromosome is the genetic material. It looks like this. I showed you the karyotype. Mesosome is present in another structure present in bacteria. So the correct answer is nucleosome. So now we come to the end of the 30 questions. And yeah, I have given you 15 questions extra for your practice, and all the answers will be shared with you for these 30 questions, and the 15 extra questions will be shared with you. If you have any doubts, you can open your mic and discuss with me. So let's consider it as no doubts, or questions you can discuss with me. You can open your mic. Malpe College? Anybody? Hosangdi College? Anybody, do you want me to clear any other concepts, or you have any doubts in what I have explained? Koteshwara? If there are no questions, then I think we'll wind up today's session. So thank you everyone for your patience, and please read through it, and if there is any doubts, please ask. Okay, thank you.